Hello, everybody. Welcome to Miss McGuire online video lectures. And today we continue covering gene action from DNA to protein chapter. And this is human genetics. So in my previous lecture, we talked about protein synthesis. And proteins are essential to your life. Actually, not just your life as a Homo sapiens, but to a life of any single living organism on this planet. So I'll give you example. Your heart is made of muscles, cardiac muscles. Muscles are mostly made of proteins. So those proteins of your heart, they contract and pump your blood through your blood vessels. So no proteins, no heart, no contraction of the heart, so blood cannot move through the blood vessels. And even if you look at the composition of your blood, inside your red blood cells, you have protein called hemoglobin. Hemoglobin carries oxygen through your body. So no protein, no hemoglobin, no protein, you cannot carry oxygen, so your tissue, your organs will die, right? So diaphragm. Diaphragm is skeletal muscle, mostly made of proteins. Those proteins contract, allow you to breathe. So no proteins, you cannot breathe, you will die. Your digestive system, to digest your food, to break your food into nutrients that your cells and your tissue, your organs can use, you need enzymes. Those enzymes are proteins as well. So no proteins, no enzymes, you cannot digest food. Those nutrients will not be delivered to your tissue and you will die. Okay, so that's us, human beings, right? But even if you look at small organisms such as bacteria, such as amoeba, plants, some other animals, fungi, neither one of these can function without proteins. And you constantly need to synthesize the proteins. Your cells synthesize proteins. And to make proteins, to make functional hemoglobin, to make functional proteins, actin, myosin, that are important in your muscles contraction, to make enzymes, to make protein hormones, you need instruction how to make those proteins. And this instruction is stored in your DNA. So we go from DNA, the piece of DNA that codes for particular protein is called gene. So we need to make a copy of this gene first, and this process is called transcription, and we make messenger RNA. Then from messenger RNA, ribosomes in your cells gonna read that messenger RNA and make protein, follow those instructions that now are in messenger RNA. And this process is called translation, right? And that's what we discussed in our previous chapter. So you can see on this diagram, we have messenger RNA, we have ribosome, and those like tails hanging down from a ribosome, those are polypeptide chains that will be a protein. But it's not enough just to make protein. We need to fold each protein into its precise 3D shape. And folding proteins into 3D shapes called conformation, right? So if proteins are not folded correctly, if they then they cannot function. And if proteins not function, I just told you what's gonna happen. You cannot breathe, you cannot eat, your heart cannot contract, your blood cannot carry oxygen, and actually neither function is possible. No one function is possible inside your cells and inside your body without proteins, right? So now proteins have several levels of structures. So first is our primary structure. This is just the chain of amino acids, because if you remember, proteins are made from amino acids. Then we have secondary structure, tertiary structure, and quaternary structure. So this is just review of a previous lecture. And here you see the primary structure on the top, a sequence of amino acids. Secondary structure is an alpha helix or beta pleated sheets. Then we have a tertiary structure. And if protein is made of more than one polypeptide, so that's one polypeptide. And some proteins just have tertiary structure, and those are they're already functioning proteins. But bigger proteins, they might combine several polypeptides. Like in this example, we have 4 polypeptides combined, making one functioning protein. Right, so now I already covered this part, but now what's gonna happen if protein misfold, right? But first, when protein begin to fold, proteins begin to fold after the amino acid chain winds away from the ribosome. Remember, ribosomes make this protein chain, makes protein that made of amino acid chains, and when it's when the synthesis is done, when primary structure is formed, that protein, that amino acid chains disattach itself from ribosome. And then proteins begin to fold. Some proteins do not fold properly, but luckily you have mechanisms inside your cells to destroy those misfolded proteins, because you don't want proteins that are not functioning. And actually, some misfolded proteins can cause diseases and even kill you. So if we have misfolded protein, your cells attach like a red flag to those proteins. So they are tagged with the special small protein called ubiquitin. So ubiquitin is a small molecule of protein that is like a red flag that's gonna tag misfolded proteins. Now, protein with more than one tag is taken to proteasome, a tunnel-like multi-protein structure. So look, even this is a protein, proteasome. The ubiquitin is a protein. So other proteins help you to get rid of misfolded proteins. So when the protein is tagged with ubiquitin and is taken to proteasome, then protein moves through this tunnel. So proteasome is tunnel-like structure. So protein moves through this tunnel and it's straightened and dismantled. So it's straighten it and break it apart. Proteasomes also destroy properly folded proteins that are in excess or no longer needed. So here's your mechanism that include ubiquitin and proteasome that allow you to get rid of those proteins that are either misfolded, or you have too much of them, too many of them, or you don't need them any longer. Right? So here are the models that show us this process. Here's our misfolded protein. This is our ubiquitin that is attached like a little red tags, and is taken it to tunnel-shaped structure, proteasome. You see how protein moves through. It became straightened up and broken into smaller pieces, first peptides, and then in amino acids. This is called protein degradation, and you can just recycle and reuse this amino acid, hopefully build now correct proteins that will function, right? Okay, so that's a mechanism that prevent your body from accumulating proteins that are misfolded. However, as any mechanism, it doesn't mean it's always gonna work, right? So that's why we have diseases of protein misfolding. Now, protein misfold from a mutation or by having more than one conformation. Mutation alters the attraction and repulsion between parts of the protein, and that's why it cannot be folded correctly. And then we have special proteins, prions. Well, you probably heard about them before. Those are ones that cause mad cow disease, scrapies. It's really a pretty dangerous protein when it's misfolded. But normal protein, prion proteins are useful, especially they very you use them in your nervous system. You find higher concentration of prion proteins in your brain, spinal cord, and lymph nodes. So prion protein is a normal protein, but it can be folded into several conformations. And some of these conformations are dangerous. And then prion protein became just a prion. And that's a prion that causes diseases such as mad cow disease. Moreover, if prion protein became a prion, misfolded protein, it can be passed to other proteins upon contact, propagating like infectious agent. It can turn normal proteins into prions. In several disorders that affect the brain, the misfolded proteins aggregate. And when they aggregate, they destroy your nervous tissue. Different proteins are affected in different disorders, however. So let's look, that's why it's absolutely important for proteins to be folded correctly. So here you can see the whole list of diseases that we believe are caused by misfolded proteins. Alzheimer's disease, familial amyotrophic lateral sclerosis, frontotemporal dementia, hereditary ATTR amyloidosis, Huntington disease, Parkinson disease, Lewy body dementia, PKU, and prion diseases. All by Huntington diseases are genetically heterogenic. That is, abnormalities in different proteins cause similar syndromes. So you can see some of the diseases like Alzheimer, Parkinson, dementia, look, it's different type of dementia, prion diseases. Even if you didn't take any biology or anatomy, physiology classes, you heard about those diseases. They are really very devastating, and they are caused by different misfolded proteins. We will talk a little bit more about this prion disease. It's a subgroup of protein misfolding conditions. They first described in sheep, which develop a disease called scrapies. More than 85 animal species develop similar diseases. Prion diseases in humans are caused by either a mutation in a prion-related protein gene, so it's PRPN, that's a protein-related, prion-related protein gene. So on the chromosome 20, you have this gene that have information how to make normal prion proteins. So you'll have normal prion proteins. But if you have mutation in this gene, you might make misfolded prion proteins that became a prion, and they can cause disease. Or, so it's either mutation in PRPN gene or transmissible prion protein, sporadic Sporadic means it just developed without any prior conditions. Thankfully, prion diseases are pretty rare, right? So that's good for us because there is no cure for prion diseases. So here are prion diseases in humans. And some of those name of these diseases, of course, like so complicated, and I wish they just change it because they used to call diseases after scientists, they describe them, but now they're trying to get rid of these complicated names. But the first is Creutzfeldt-Jakob disease. Again, that's all prion diseases. When normal proteins became misfolded and they get aggregated in the brain and spinal cord, and they pretty much kills neurons that are cells of your nervous system. Now look at these symptoms: memory loss, nervousness, trembling hands, blank face expression, jerky movement, unsteady gait, personal changes, anxiety, depression, impaired thinking, insomnia, visual loss, difficulty speaking and swallowing, coma with death from heart failure, pneumonia, or infection within a year. So really, really devastating condition. And the worst part is that we don't have any cure. Fatal familiar insomnia. It's a progressive inability to sleep and vivid dreams during scant sleep. Physical and mental deterioration, coma, death within a few months. Gertzmann-Straussler disease, memory loss, dementia, ataxia, uncoordinated movement, amyloid plaque in the brain, and death within 7 years, right? This give you 7 years of suffering before the patient dies, right? So, and again, why we covering this part of the of the proteins? Because if you look at your objective for this lecture, you need to explain why proper folding of proteins is important. And if you know that proteins that are misfolded can cause such devastating conditions that people die within a year, within 6 months, within 7 years, then you know the importance of proteins and their proper three-dimensional shape, right? So here's some other condition of prion diseases. So the first prion disease recognized in human was Kuru. It's affected the native poor people who lived in the remote mountains of Papua New Guinea and was traced to a ritual in which the people ate their war heroes. Now look, people and any animals should never eat other animals' brain and spinal cord, and especially never eat brain of your own species, because the brain and spinal cord and lymph nodes is where most of those prion accumulate. So if these people, why they had this Kuru disease? Because they ate brain of their war heroes. So I guess they believe that they became stronger and strong and brave like those heroes. Another, we already talked about this Creutzfeldt-Jakob disease, and it's actually the one that is called mad cow disease that also can affect cows and people. And the cows get this disease because of, guess what? They also were fed with bones of, they added the bones of, well, they made like a, I would say like a flour, right? Like a flour from a bone. of other cows. So the cows ate the spinal cord that was inside the other cows or part of their brains. So that's how this mad cow diseases first developed and first was discovered in cows, but we know people can have it as well. Now, but how prions cause the disease? Prions cause disease both by spreading the alternative form and by aggregation of the protein. So those prions, they can make other prions misfolded, and they can aggregate. The normal function of prion protein is not well understood because we do have normal prion protein, but the normal prion protein can misfold into prion, and it can alter the form of other prions, and it can aggregate itself in the brain and spinal cord. But normal prions, they are needed. We have a gene to make prion proteins, and we believe they're important in creation of synapses between neurons in our brain. So it's important also in the protecting the nervous system and forming synapses. All prion diseases are diseases of protein folding. The infectious form have beta pleated sheets in place where the normal counterparts have alpha helices. So remember, proteins can have this shape of spiral that's called alpha helices, or like this, the beta pleated sheets. So if we have a normal prion protein, they have alpha helixes, more alpha helixes. If we have misfolded prions, they have a lot of beta pleated sheets, and it's allowed them to aggregate, to stick to each other, and that will cause disease. So here you can see neuron, because as I said before, prions are abandoned in the nervous tissue and lymph nodes. So they kind of like look inside the cytoplasm of a neuron, and we can see that we have a normal PRP, that's normal prion protein, and we have the protein, see, like this C shape. This is prion protein SC, scarpies. It has a different shape. So original scarpies PRP molecule converted the normal prion into scarpies. So here we have scarpies and a normal, and then this original scarpies converted a normal protein into scarpies. And then they converted more and more and more prions into, we call this prion proteins, or normal PRP. They convert them into scarpies, PRPs, and then they can stick to each other, forming aggregation of prions that finally will destroy this neuron. And if neuron is destroyed, then the nervous tissue is not functioning, and if your brain and spinal cord is not functioning, then your body pretty much not gonna survive this type of damage, right? Okay, so this is our last slide, and I just want to remind you that we discussed the importance of protein folding and diseases that are caused by misfolding proteins, and specifically prion diseases. Okay, so thank you very much for watching, and I hope it was helpful.